Bismillahirrahmanirrahim, and hopefully everyone is fine. Have a good day. Let's start with our chapter 4. We should continue from the last time we stopped at cell culture. This one is one of the studies that I have used. I have performed with my lecturers quite years back. So the idea of this paper is to show you what are the applications available when it comes to optical based system or microscope. So in this study we will look at what we call high resolution imaging of biomolecular binding studies using a system called surface Plasmon microscope. So the standard system is called surface Plasmon microscope, but what we have used is the wide field surface Plasmon microscope. Alright, let's see the definition a little bit. So the system, the surface Plasmon SPR, what I call the short form is SPR, surface Plasmon resonance based microscope, built via Plasmon configuration. That means it's a prism base. So where you produce an image of a sample via intensity map, where the intensity of the image depending on the light which is coupled to the surface Plasmon. But the problem is that you have a reduction in the lateral resolution of this system, and because this Plasmon they propagate along the surfaces. Therefore, in this system we came up with a wide field surface Plasmon microscope. So we no longer use the prism. I'll show you in the diagram. So it is no longer used as a prism base, but we've used objective lens. So like optical based system, still optical based system, but we have replaced with the what we call high numerical aperture objective lens, and then in this study we have looked at one is micro pattern extracellular matrix proteins. That means the bindings of antibodies to antigens studies, biological studies, right? Now this is the what we call the Plasmon based configuration. That means you have this is the normal prism, okay, and then this prism is coated with metallic layers of materials. So for example here what they use is a silver or metal film, approximately 50 nanometers. On top of that material is what we call biological compound, which is microstructured on the metal layer. So you will have an incident beam at a specific angle. At this specific angle you will achieve what we call, you will trigger the free electrons on the surface, and then there will be charges from electrons to electrons, so that it will oscillate the free electrons on the surface, and then eventually there will be energy decays. But the reflected light is what we call recorded, and you have the intensity map projected from this reflected beam. So this is the what we call the traditional or the old surface Plasmon system, the earlier one. So as we said before, the surface Plasmon, it propagates along the surface. So therefore, when you have some sort of a structure or biomaterials on the surface, so it's gonna show the mapping of how the attachment took place. So it will be nice to show if you can find out more papers on the surface plasmon. So how they image a cell. So if you have a cell sitting on the surface, so you will have the propagation of energy from free electrons on the surface, and then it will be reflected back. So this is the incident light and then reflected back. This is the system that we will use that on the wide field surface plasmon system. So this figure number 3, this is the objective lens. Then we have oil. So this oil means the lens is oil immersed objective lens. So you have the oils actually touching the glass side, and then you have the 0.1 mm thick substrate, and then you have the coatings of gold on chromium. So in order to make the glass, in order to make the gold metallic layers to stick onto the glass, so you need 1 nanometers of chromium. That's the function of this chromium. Chromium will what we call make it so that the gold will stick strong enough onto the glass slide. So you have glass, chromium, and then gold, and then you have the proteins being deposited on top of the gold layers. So the beam direction is from the underneath, so we are actually imaging from the underneath of the samples, and then there will be oscillation of surface plasmon, and then when the light will get reflected back, and it is further processed. Now from this theory, I want you to understand how the optical system works, right? So how the optical system works in both the standard and also in the wide field surface plasmon system. So figure number 4, it just shows you the gold, chromium, and glass. As I mentioned before, the chromium function is to make the gold stick strongly onto the glass. So, and then you have the site where we deposit the protein. So just remember, the site where we deposit the proteins or extracellular matrix protein or the cells is on top of the metallic layers, not on the glass. It's on the metallic layers, right? Another technique that I would like you to explore also is called micro contact printing. So in the micro contact printing is actually we use PDMS stamp. I'll show you this. Yeah, this is the image. Okay, let's have the full figures of right. Okay, so at the beginning, this is I'll explain from the fabrication of the stamp. So you need a glass template. I mean the glass template with the grids of patterns. It depends. So in this study particularly, we used 1.8 micrometer size and also 3.8 micrometer size. So two types of PDMS stamp, polydimethyl stamp, PDMS stamp. This is the glass template. So at the beginning you want to cast the stamp, so you put the silicone elastomer on top of the glass template, then you let the PDMS set first overnight. After that, the next day you remove the PDMS stamp, right? And then you use that PDMS stamp with the different sizes. So in this study we use two sizes, 3.8 and 1.8 micrometer. So you ink the stamp with the fibronectin solution. For example, in this study we use fibronectin extracellular matrix protein, and then Now what will happen is that when you remove the stem, the excess of the fibronectin will stay, and then only those which is attached onto the stem will remain. Now this stem you can use to stamp the surface. You use the PDMS stamp, which is ink with the protein, to stamp the surface. You press the PDMS stamp onto the glass, the substrate or the surface plasmon substrate, for example, for 30 seconds, so that you make sure there is protein being printed onto the surface. So when you do the first print, it's going to look like this. So you have in one direction, and then this is the 3.8 micrometer size, and then you have the second one is the other way. So you have it cross-linked pattern. So that's what, when we took an image on the system with the normal microscope, optical based microscope, so you can see in two directions. One direction, this one presumably is the 1.8 micrometer line pattern, and then the other way is the 3.8 micrometer. Now next is we will do an analysis of binding event using the surface plasmon, just to show that the system, surface plasmon system, is sensitive enough to what we call, the system is sensitive enough to identify the differences between the binding of antibody to the antigens and also to the proteins. So yeah, this is the complete studies that we have performed. So I want to show you in the full flow diagram. So let's say first, as I mentioned, we have deposited the fibronectin surface onto the surface. So one, and then the free areas is being filled by another protein which is called laminin. Both are extracellular matrix protein. So we have put two is to show the ability of the surface plasmon to detect the changes between detection of binding event between these two proteins. So because it is an optical system which based on the molecular species being deposited on the surface, so we would like to test the system whether the system are able to detect changes in the layers of materials being deposited onto the surface. So this is what happening now. So why laminin is in between the fibronectin? Because a protein will not attach to another protein, that means overlapping. So the protein will fill in between the free areas which is not printed by the micro contact printing. Right, let's look at the diagram and also the two images. So before I start, these images, the black and white in number one is all these surface plasmon images, white field surface plasmon images. The next to the surface plasmon is AFM images, and this is the process, step-by-step process. So why we use AFM? AFM is called known as atomic force microscope. Atomic force microscope is also an optical based system, but it produces the results, imaging results, which is close to the sample in nanometers as well, and it function with the lasers being positioned on top of the cantilever tips, and the tip will scan onto the surfaces. So that's atomic force microscope, atomic force microscope system able to produce topographical outputs of images. Therefore, it is quite ideal to be compared with the surface plasma system, because surface plasma system also detecting or measuring the molecular species which is bound to the glass substrate. Right, let's look at the first image. So initially you can see very low contrast image, right? You will see not much of the pattern or the lines. It's not visible. So why? Because it is in only very small molecular, I mean very thin, very thin layers of monolayers of fibronectin being deposited on the surface. Then you can see in just one direction. But as soon as you fill in the free space area with another protein, what happened is, so this is what happening. So initially you have one layers of protein, now you have another layers of protein attaching to the surface. Therefore you can see that in both direction you can see the patterns become more visible. Similarly also with the AFM system. Now are the surface plasma system really sensitive to detect molecular binding onto the surface? So what we did is we tried to further amplify the attachment process. That means now it shows that from low contrast it become little bit high contrast at this stage, the second stage. So can we further increase the contrast? Yes, of course. So we attach the laminin with laminin antibody, rabbit anti-laminin antibodies. So and then we couple them onto the laminin, process it, and then now you can see the contrast is getting higher or much increase the visible of the patterns, right? Okay, and then when, so now is a good way to test where we have to focus on the fibronectin to show whether the attachment on the fibronectin now will reduce the contrast. Yes, it does. So when we put the mouse anti-fibronectin, that means antibodies coupled to fibronectin, so you can see the patterns become less visible. So this is the same substrate image with AFM. You can see, because the layers become more or less same thickness. So in this one image also actually there is a reduction, but this is the images which shows qualitative results. So we did also some processing with this all these images. So we managed to do some measurement of quantification. So in numbers we can actually see the increase and reduction of the contrast. Last but not least, so we try to bring back the laminin into the process. So we further what we call amplify the rabbit antibody by using goat anti-rabbit antibodies. So now we treat again the samples, and then we can see there is an increase again on the pattern. So similarly in AFM also the patterns become visible. Hopefully I will have one diagram here to show. Yes, this one. Okay, let's see. Now so this is the sample that's been imaged with the how it looks in the surface plasma system. So you have fibronectin free area, fibronectin patterns. That means the square shape in the middle, the lines is the fibronectin area, and then the fibronectin free area is the square one. So this is the patterns it looks when you scan with the AFM system. This is what I meant by topographical images. AFM are able to produce topographical images. That means they are imaging from the top to show like a scanning system, which will give you high resolution imaging as well. As we focus in this study, one of the application which is very much in interest in the field of biomedical is high resolution imaging, and therefore surface plasmon is another option, and is also come in hand with the, can be compared by using the AFM. But AFM have certain advantages and disadvantages, so later we will look at AFM, what we call AFM advantages and disadvantages. Okay, this is what the quantified result looks like. So we have managed to record the contrast difference in one, and then according to the process that I have explained before. This one, at the beginning, the contrast is very low at when we have fibronectin protein only, but when we mix, we have the laminin coming in between the free areas, so we have the contrast increase little bit. And then when we record it again, the process of antibody for the laminin, so now the target, the molecular species deposited onto the surface becomes much higher. Okay, and then when we focus on the fibronectin area, it further reduces the contrast when we applied the antibody for the fibronectin, and then when we targeted again the laminin area, so the contrast increase back to, it becomes higher. So this is what we call the quantified measurement, which will complement the qualitative results that we have produced earlier. So from that findings, it shows that this one is just the images to show the printing patterns achieved by micro contact printing, just to show that the process is working nicely. And then ABCD is the fluorescent based studies on the antibodies, just also to show that the antibodies is functioning as it should do, right? Now from this study, we know that wide field surface plasmon system can be used to study the attachment of molecular species onto the surface. That means the deposition of proteins does show very sensitive recording by the surface plasmon system. And then from this study, it shows that the surface plasmon system are sensitive enough to pick up signals in less than a half micron. So from the measurement, it shows that the resolution can be pushed down to half a micron. It's not same. It's not same with the AFM system. AFM system, it can detect also down. I mean, pushing the resolution down to nanometers. But as I mentioned, AFM shows topographical images. That means the view is actually from the top. The view is actually from the top, and surface plasmon is looking from the bottom. That means the layers which has been deposited, as I show the orientation of the objective lens. Now from this study, it shows that surface plasmon system can and able to detect the molecular species binding onto the surface. From this paper, I would like you to also explore another application or papers, studies which we did on the cell imaging. this paper now is on what we call protein binding. So I want you to find out papers on cell imaging. See, it's the same work I did back in I think 7-8 years back. So you try to find out another studies which to do with the cell imaging, as this will help you to understand the cell culture process which is explained in the previous lecture. So now find out cell imaging paper using wide field surface Plasmon resonance microscope. So that's the thing that kind of homework for you to do to find out about these studies. And then I have one question to ask to the student. So I have one question. Right, this question is just to make sure that you actually understand the lecture, and I just need a quick answer from the students. So in these studies I have explained to you micro contact printing. So sometimes it is also called micro surface patterning. So in micro contact printing, I want you to answer from this paper that what are the materials that been used to create the stamps that been used for micro contact printing. What are the materials? The name of the materials that been used to create these stamps. So write your answer in the comments. So I will check it out later on. So hopefully this is one of the application chapter for biomedical engineering application subject. So hopefully you will explore some more. Remember, find out the papers about the cell imaging using wide field surface Plasmon system, and it is a paper you definitely will find. And also I will also share in the next lecture on regarding how other optical system works and function, so that we can compare one or two system in this subject. So thank you very much, and hopefully to see you guys in the next lecture. Have a nice day. Wassalamualaikum